Good afternoon. Welcome. This is the 12th year that we have had this course in demystifying medicine. The point of it all is, quite frankly, to excite your interest, not to fill your heads with all sorts of facts and numbers. So particularly for students and fellows, listen and think, but you don't have to write everything down because nobody's going to quiz you about it. However, if you attend and sign in for 50% of the 20 sessions, and at the end we have a electronic multiple choice exam, which you can take as many times as you need, and you pass it, and you'll get a certificate which says that you took the course, which I am informed is of some value. There are roughly, I think this year there are roughly 850 people at NIH who are signed up for here. There's about maybe 100 here. Several hundred watch live. It's video, it's live cast on the NIH campus. If you're here, you can ask questions and take part in the discussion. If you're there, you're somewhat limited. But then after the third day, it goes into the NIH video archive, and it goes around the world. And there are literally thousands of people who watch this. It's in 18 countries. They use the material that we generate. And right now, there are better than 24 major North American teaching institutions that do too. So why do they do that? Well, because what we are trying to do is to cover the big gap in more or less 20 major human diseases each year, and it varies from year to year. So where possible, we have a live patient who, in their own words, tells you what it's all about as they perceive it. And then a sort of translational physician scientist who discusses the disease. And seeing as many of you, have not had things like pathology or histology, I discover there are even some postdocs who never had biochemistry. So there's a lot of heterogeneity. And so this is not an intensive clinical grand rounds where it's directed in that way. And then the second speaker is a basic scientist who kind of summarizes what we know, don't know, and should know. And both of those individuals talk about why they're here at the NIH, spending their lives and efforts. What do they work on, and why is it important? And throughout all of this, we encourage you, even while people are talking, if somebody talks and uses terminology or something you don't understand, make a noise, stand up, say something. This should be as relatively informal as possible. Now, if you sign up, which I guess most of you have on the listserv, you'll find every week there'll be a short introductory description of what the topic is and sort of some thoughtful highlights. And then the CVs of the people who are going to speak, a few selected references, which we hope you'll get a chance to look at some time, because they're really quite selective and usually including at least one recent review. And then all the PowerPoints are either up before or after. And if you go to the video archive, you can see talks and PowerPoints for 12 years worth. So it's kind of interesting and important. And at the end, there's plenty of time for questions, discussions, and so forth. Okay? So does anybody know what that is? Do you know what that is? Yeah, it's the Brooklyn Bridge. So why do we show a picture of the Brooklyn Bridge? Because that's the ideal logo for what we're trying to do. So many of you who are very expert in reductionist science are over there in Brooklyn. And others who are more clinically involved, clinical trials, translation, whatever it happens to be, we're over here in Manhattan. And so the whole idea is to get us to talk to one another, to listen to one another, to communicate. And that's what these two guys are doing. They could walk out on the catwalk, and that's kind of symbolic of where we are. Now, this isn't just fairy tale. I think this is the major problem in biomedical science today, is that people are experts in little parts of the pizza pie, but they can't communicate with the people from the other parts. And so we lose the big picture. What's important and what isn't? And what do we know and what do we don't know? Also, the picture was taken by my grandfather, so I have a vested interest in it. Okay. Then how do we get to the next one? So what's aging? I try to, when we pick out these things, I sort of sit down and meditate a little bit, try and figure out what do I know about it? And I figured many of you know much more. But, you know, is aging something like this terrible poem by Oliver Wendell Holmes, The Deacon's One-Horse Shay? This was a one-horse carriage that was built of the purest things that could be found, the best. Now, how did it end? It went to pieces all at once, and nothing first, just as bubbles do when they burst into the wonderful one-horse tray. Now, is that what aging is about? Does everything just sort of give out? Is there a basic behind-the-scenes biological phenomena of aging? But people rarely do that. They usually, although they may be much older these days, and everybody's getting older these days, but even, you know, as you'll hear more. But when you get older, lots of things happen. Degenerative diseases of the nervous system, the cardiovascular system, everything. So is aging the same thing as the degenerating diseases? Are the degenerating diseases the same thing as aging? This is a very important question, whether there's a common underlying pathway behind all of this. At least these are the things I wonder about. Are the degenerative diseases, whether it be Alzheimer's or cardiovascular, are they increasing because the population's getting older? Or is there some other thing? And then most importantly and excitingly, are the experimental biochemical and molecular mechanisms which prolong life in invertebrates, mice, and maybe humans applicable to the human being? And how can you study all this? So today we are really most fortunate in having two truly global experts in not only this, but the whole parameter of what is aging. So I'm going to very briefly introduce both of them to you, and then Dr. Hodes will be the first speaker. So Richard is the director of the National Institutes on Aging. He's a widely recognized immunologist, a physician, graduated from Harvard Medical School, trained at the Mass General, trained here in oncology at the National Cancer Institute. He's a member of the Institute of Medicine, and he presides over this whole arena that is encompassed by the National Institutes of Aging, which covers much more as we will hear, than what I put there. I just discovered several months ago that, for example, Alzheimer's disease, the major focus for it is in the National Institutes of Aging. And Richard is the coordinator, the director of all of that. So he's going to speak first, and then following his talk, Toren Finkel from the Heart Institute is going to discuss at a more reductionist level his work in relationship to mitochondrial metabolism, turnover of cells, and the aging process itself at a more, let's say, basic level. Now, Toren is also a physician. He also graduated from Harvard and has a PhD in biophysics, and has been here at the NIH for not too many years. But he's chief of the cardiology branch of the intramural research program in the Heart Institute. So, Richard, oh, wait a minute. Whoa. Come. Take your thing and we just sit down. Okay, so, okay, yeah. So this is our class, Lila. And so this young lady is a very dear friend and a most remarkable individual, aren't you?
Well, if you say so.
Right. Now she is, how old are you?
95.
She is 95 years young. And how old is your sister?
She was 100 this past August.
And how old were your parents?
86, each of them.
And what do you attribute your youthful appearance and vigor to at this young age?
I really don't know. People ask me, what is my secret? I wish I had something to say. I have no idea. I never thought of being this age. I really don't know how I got here, but it just happened.
So what do you do? What is your, do you have a occupation, profession?
I retired I was at Howard University 43 years. I retired in 1991. I've been busy in the studio since, and in 2006 my husband died, and so now I'm the housekeeper, the one who goes to the grocery store, the one who goes to the cleaners, as well as doing my work in the studio.
Lila is an accomplished artist whose work has been shown in many institutions in this area and elsewhere. Did you ever do much physical activity?
Well, I was not really terribly athletic as a child, but I did, believe it or not, at my height, play basketball. In high school, girls' rules were very different in those days. And so I was, as short as I wasn't as short as I am today, but I was still not very tall. But I played basketball. I got somebody's elbow in my face that loosened my teeth. But later on, I didn't do anything special until I somehow got into the class at the Bethesda Hospital, Navy, where they had a wonderful pool, and they had an exercise group, which somehow I found out and joined, and that was about the 80s. I used to go there. We had a group that exercised. Just one of us led the group very informally, and then we'd swim. And I did that, and we all did that until they took down the pool.
But I think you told me this was about for 30 years, right?
I suppose. We figured it was sometime in the 80s that I started, and I really don't know when they took down the pool. They took down the whole building.
Oh. But you swam 36 laps, right?
Well, I got up to 36 laps. 18 is a quarter mile. And so when I was feeling good, I could do 36.
Do you smoke?
Never.
No bad habits. That
Oh,
you
well, I
can
wouldn't say that, but I don't, I don't smoke.
Have you had any other health problems?
Uh, well, I had cancer operation. I had a section of lung removed. I have carcinoma. Why? I have no idea. Say, I never smoked. My father smoked cigars, and I always sat behind him in the car when we drove. I was the one who sat on that side of the car. My first husband also smoked cigarettes until the children were small, and then he realized that he didn't want the children to smoke, so he quit. And I never did.
So when was that you had your part of your lung removed?
206.
It's almost 20 years ago.
Good. Yes.
And believe me, it's hard to keep up with her when you're walking along the street,
Yeah.
right?
Well, my balance is bad, and I do better faster than I do slower.
Is there anything you'd like to ask the group?
I don't know. If you tell me what makes some people get older, I'd be delighted.
Maybe you have any questions, a few that you'd like to ask Lila. Anybody? Yeah, speak up. Just,
What was your diet?
what was your diet?
Well, I think I do. I do eat, being aware of a good diet. I don't do anything particular. I was a chubby child, and from the time I was about 16, I was aware of diet. So my father was a doctor, and I would complain that I didn't lose weight, and I only eat so and so and so and so. And he'd say, "Your system doesn't require that much food," and that was it. And so I've been watching my diet or aware of my diet all these years.
Lila, do you take food supplements like lots of vitamins?
Oh, well, I take general vitamin, and here lately my doctor has told me to take extra vitamin B. I have vitamin B12 shots. It's supposed to help my balance. I don't know that it does.
Yeah. All right, anybody else? Okay, Luba, speak up.
Lila, many people, when they retire, feel that they kind of not needed and bored, and retirement sometimes not quite agrees with longevity. How active you are socially and still in your profession, and how much of that helped you to go with the last few years?
Well, I don't know. Doing artwork is about the only thing I know. I don't get to the studio every day. I get involved in social things, and as I say, I'm in charge of the house, and I have to call the plumber or whoever is needed. But art is the only thing I know to do. I don't know what else I would do.
Lila lives in a 3-story house, and her studio is both up and down, and she's up and down those steps many, many times, right?
Yeah.
We have to congratulate you.
Well,
Well, it was just a month or so ago that Montgomery County gave you an award for an outstanding lifetime of creativity, and you were the
yeah.
awardee of the year.
Yes, it was
That's
very nice.
wonderful. Well, listen, thank you so much.
Yeah.
Sit down.
Okay.
We have to turn that off.
Well, here.
Okay. Richard?
Well, it's a hard act to follow. It's really inspiring to all of us. I'd like to talk very broadly about a few aspects of aging. I'd like to begin by giving you a really broadest picture at a population level of what is happening to the aging globally and to this country. Move from there to talk about some of the clinical issues that are related to aging and some of the progress we've made in addressing them. And then finally talk about some of the underlying processes, biological, molecular level, that are associated with aging, which I hope will be a real transition to what you'll hear in terms of an expert perspective on one important aspect of molecular science. So to begin, this is, I think, one of the more graphic illustrations of what's been happening to the population of the world in terms of its age demographics. So what you see plotted here is from years from 1950 to 2050, two lines, the proportion of people, percentage of the world's population that are either older, that is over 65, or young, under 5. So if you look on the left, 1950s, 1960s, there were about 3 times as many kids under 5 as people over 65, about 15% to around 5%. And then look dramatically what's happened until we reach the current point at which the percentage of young goes down, percentage of old goes up. So this is really fairly historic. We're approaching undoubtedly the first time in human history there'll be as many people at older age as there are younger. And if you look at what is projected by 2050, that's going to be completely reversed. So quite apart from all the medical issues, the sociologic implications of this are huge. It's just going to be a very different world. And this is perhaps a background for understanding what a very dynamic point we're at and why it's important to understand what aging means to us societally, medically, and in terms of underlying biology. The theme that we'll try to undergird through here is that aging is a major risk factor for most chronic diseases, and an evolved hypothesis that this is because the biological process of aging itself, multifactorial as it is, underlies some of the common risk factors for multiple diseases. So you can see scattered around aging here, changes in the immune system, stress, genetics, DNA repair, metabolism. Certainly mitochondria deserves its own circle right here. But the notion is that all of these changes which happen with age might be common risk factors for disease. And so as we'll see, thinking has been that in addition to trying to treat or prevent individual conditions, if we can address some of these conditions which may be underlying and in common to multiple diseases of aging, we may be even more effective in trying to improve health overall. So this is just a slide to show you the many, many conditions illustrated here, how age-dependent they are. So whether it's kidney disease, stroke, congestive heart failure, osteoporosis, you can read them all. The graph shows here how dramatically they increase with age. And it's really a fair question whether this is simply an accumulation of stochastic or random events that happen as time goes on, or whether there are underlying biological changes that occur that are responsible for these. And we'll talk about some of this. Now, interesting, I think, is what's been happening in trends for longevity, for aging in this country in comparison to the rest of the world, and for subsets of our world population. So this is really some socioeconomic or societal commentary. What you can see here for men and for women from 1920 to 1950 is life expectancy from the age of 50. So this is how well people are doing if they make it to 50 and then beyond. And it is shown as a function of the richest to poorest 10% of the population. This is really quite telling. Look at men first. You can see where we were in 1920, where the richer people did better, lived longer. Not only is it true now, but what you can see is this is through 1950, and it continues. The gap is continuing. So the socioeconomic advantage or disadvantage of being rich or poor in this country, and this is more dramatic in this country really than most any other part of the world, is illustrated here. Now for women, it's in fact even more dramatic, so that if you see here in the last years, the poorest 10% and 20% of women have actually not gained. They've lost in terms of life expectancy. Some of this has occurred at earlier ages in life, but it's also true at older ages. The fact that it's socioeconomically related indicates that it is in large measure likely to be manageable, malleable, changeable as a result of factors that we can do something about societally. If we take a look at how this compares worldwide, this is, as of 2014, the life expectancy from birth. Japan had the highest at age 84. We're not first, we're not second, we're not in the top 10. We're down here, a poor 34th, below a lot of countries who, in terms of socioeconomics and other parameters of wealth and well-being, ought to be at a disadvantage. So this is about as much as I'll say here in this talk based on medicine, biology, but it's a challenge to all of us to understand what's happening in this country versus the rest of the world. A lot that can be done that needs to be done. If we look at the same issue here, this is a plot saying something very similar, but it's showing the U.S., this is the bottom line, versus comparable country averages, that is, socioeconomically comparable countries. In 1980, we were a little bit behind. once again, this trend, disturbing as it is, the gap continues to expand. So we continue to do less well in recent years than countries of similar or even lesser socioeconomic development and wealth. So let's move to look at some of the causes of death, in fact, as of a couple of years ago. Not surprising, heart disease first, cancer, respiratory disease, you can go down this list. And I'll use this as a takeoff to describe some of the improvements we've made, and some of them have been very impressive, very dramatic in terms of addressing these individual diseases. And as I said, once we get through some of this clinical material, we'll turn back again to ask how the biology informs what's happening and informs hopefully what all of you in your careers to the future are going to be able to do to help do something about all of this. So some of the clinical research that has been carried out at or supported by NIH, which has made a very big difference, I'll illustrate here. This all has advanced our understanding of the conditions that underlie disease and also our ability to do something about it. So let's look at a few examples. This study reported just a couple of years ago is one of the potentially most informative in terms of giving us an intervention that can make a difference in life and quality of life. All of you well know that blood pressure, high blood pressure, carries a risk of premature death. It's already some years ago when the first studies were done, which at that time, contrary to expectation, showed that if you controlled systolic blood pressure, the highest of the two numbers, from 160 down to around 140 or so, you actually had dramatic decreases in stroke, in congestive heart failure, in heart attacks. And for years, this threshold of 140 as a target has been what has been behind many recommendations. In fact, there were some who suggested that for older people, maybe 140 was too low. You know, with age, vessels might get stiffer. You might need more pressure to pump the blood around and give adequate circulation. But all this was tested in a study called SPRINT, Systolic Blood Pressure Intervention Trial, funded by NIH, which compared controlling blood pressure to a level of 140 systolic pressure or 120. This was not a case where there was going to be a placebo group where you don't treat people. That would have been quite unethical. It's already known you need to control blood pressure. But we didn't know if being more aggressive, pushing it down even further than been consistent with recommendations, would be a good thing or a bad thing. So this study, as many of you may know, was stopped prematurely. Now, sometimes studies get stopped prematurely for ethical reasons because there's an adverse effect, a negative consequence of the treatment, and it's just not conscionable to go on. But sometimes, as in this case, it became so clear prior to the scheduled end of the study that there was an advantage to controlling blood pressure down to 120 that the study had to be stopped. And so you can see here the standard treatment, that's to a blood pressure of 140, versus the intensive treatment. We're looking over time at the cumulative hazard, that is the risk of either dying or having serious cardiovascular effects. And there were dramatic 25 to 30% decreases here. So we are now at a point in terms of these studies where it's up to the research to be translated by the practice community into what hopefully has the potential to save thousands, tens of thousands, hundreds of thousands of lives and to spare many some of these cardiovascular and other severe incidents. Now, it was still an interesting point about whether this was going to hold true for all ages. Or was it, as I mentioned, that maybe older people would need a higher blood pressure? Perhaps a lower blood pressure, they would have less cerebral circulation. Maybe they would have more falls and adverse effects. So the National Institute on Aging, in this case, was particularly focused in its collaboration with the National Heart, Lung, and Blood Institute, which was the lead in this, to make sure that there were people recruited into the study who were at an older age. And the good news was, shown here without going through the specifics of each, that as a function of fitness, more fit, less fit, frail or not frail, for all of these groups, individuals, adults over 75, similarly profited from a reduced blood pressure. There was not an increase in falls, there was not an increase in adverse effects, and it's only through this kind of a randomized controlled clinical trial that we can be confident of this sort of thing. So here's a success that now needs to be translated into practice in this country and beyond. And again, as invited, if there are questions at any point, please feel free to come up with them. Otherwise, we'll just move along. This is another kind of example. No secret that smoking is a serious risk for multiple diseases, cancer most prominently, lung disease, but many other conditions. And one of the great triumphs in public health in this country has been the relative reduction in the proportion of people who smoke. but there are still many who do smoke. And so there's another kind of research, behavioral research that's gone on to see what approaches might be effective in helping people to reduce smoking. In this study, there were financial incentives provided and interesting strategies. In one case, if you remained free of smoking for a period of time, you got a financial reward. In another, it was similar, but you belonged to a group. And if the whole group succeeded, then everyone in the group profited. So this added an interesting component of peer or colleague pressure to it. And there was another set that is called commitment here. In this case, you were given money and it got taken away from you if you failed, and similarly with a group. And without going through group by group, all of those were effective. Fewer people were willing to take the risk of being given money and having it come back. They didn't like that, wouldn't even participate. But those who did, those who were willing to participate there, actually had the best effect at all. But what you can see is compared to a control group, by 6 months, and unfortunately less true at 12 months, there was some success here. Modest changes, but if you translate these potentially into, again, many tens of thousands, hundreds of thousands, millions of people still smoking, the public health implications of a small change like this can be huge. And in fact, if you look at many of the causes of death and disease today, a good proportion of the risk is attributable to behavior we can do something about. So I just will emphasize that behavioral research and understanding what it takes to make people change their behaviors is an important part of what we do. So cognition is an interesting thing, and training and what one can do to improve or alter cognitive function. So let me illustrate one set of studies here that has had really quite remarkable, again, potential applications. So useful field of view is a concept. It's really not a test of visual acuity. It's the useful field of view as a cognitive function. So as you can see on a computer screen through a variety of images, the idea is to focus on a point, no change in head or eye movement, and then pay attention to both central and peripheral things that are happening, complex events that are going on. And one can be trained to do better in these tasks. The training can have to do with speed of processing material. It can have to do with memory, and it can have to do with associative or executive function. And studies have reported, indeed, that you can successfully, with a short period of time, induce changes which will, for years, give you better outcome on those tests, on these tests. Now, I'll show you a little more specifics, but as illustrated here, these were so effective when applied to a task of simulated driving that, in fact, this test has been used as a screen for certain motor vehicle bureaus, and training in these tests is being used by certain insurance companies already as a criterion for having reduced rates. So the data are good enough, the evidence was good enough to lead to both of those outcomes. But let me show you what happens in a clinical trial. This is really a remarkable one. So people were trained for a relatively brief period over days and weeks, and then they were followed for automobile crashes, serious crashes. So the level 1 or 100% is the control group that didn't get any such training, otherwise matched for a lot of characteristics. And then the effect of memory training, of reasoning training, or speed of processing training was assessed after 5 years. 5 years after this initial behavioral intervention. And we can turn to the increasingly more effective, the two of them being reasoning and speed of processing, where if you look at the scale, where this was measured by person miles or person time driving and the number of serious accidents per time, there was a 50% reduction, decreasing by half the number of serious accidents as a result of this kind of training. And this is all cognitive training, just teaching people how to pay attention. It obviously has generalized from computer training to a task such as this, and undoubtedly to many other tasks over life. And we'll come back to some other measures and diseases and conditions progressively now, but among them is going to be the issue of age-related cognitive decline and even risk of dementia, such as Alzheimer's disease. And the very same people who've been involved in this kind of treatment are in fact being followed to see if not only is this preventing accidents, but improving cognition, maintaining reserve, and decreasing the risk of actually progressing even to dementia. We'll get to that. Preventing diabetes. Diabetes, again, one of the leading causes itself of morbidity and death, but combined with other conditions, having a huge impact on quality of life, on cost of medical care. So this was a study completed some years ago, which was intended to compare the effects of either a lifestyle intervention, which meant exercise and diet, relatively modest interventions, metformin, an oral hypoglycemic that is used to treat people, in fact, with diabetes, or a placebo. And the idea here was to treat people who didn't have diabetes, but they were at high risk as a result of chemical testing, weight, their profile of risk factors. And then to look at the effect of these interventions on the risk of developing diabetes over time. So what you can see here is, particularly to emphasize, the effect of treatment, but also the difference in effect as a result of or correlative with age. So for the 25 to 44-year-olds, relatively young adults, about a 50% reduction in new cases of diabetes with either treatment, the metformin or lifestyle. By 45 to 59, the metformin a little less effective, lifestyle still effective. And by this criterion, which I should emphasize is just the usual glucose tolerance test. It's not the only criterion, but it's a principal one used in diagnosis of diabetes. Look at what happens to the over 60-year-olds. For reasons we don't well understand here, for this parameter, metformin not so effective, but lifestyle more effective than any other group. More than a 70% reduction in diabetes. So I think probably, in contrast to what was expected, you can name your prediction here. Can't teach an old dog new tricks. People are at an advanced age going to have a much more difficult time in making a change. The result was, in fact, if anything, the opposite. At an older age, interventions that began after 60 could have a dramatic effect in preventing diabetes. So if we're looking for this kind of evidence that modest changes in lifestyle can make a difference, if we can get people to adapt them and comply, this is another striking example. Another case where a trial had to be terminated because it was unconscionable to go on. It was just so effective. And subsequently, 5 and 10 years later, it's been shown that, again, some of the positive effects in avoiding disease in these groups persist. So I'd like to spend a few minutes talking about one of the diseases that is highly associated with aging that to many in the population has become one of the most feared of diseases, Alzheimer's disease, dementia. The loss of memory, of cognitive function, the inability to identify objects, to think and reason, forgetting loved ones, the impact this has, huge. And I might just stop and ask, how many of you have had a personal experience, a family member, friend, loved one, somebody with Alzheimer's disease, by a show of hands? Yeah, and me too. It's clearly these days a majority of people. It's estimated that something over 5 million people in the country are affected today, something like 50 million affected worldwide. When the disease was named or identified in 1906 by Professor Alzheimer, who cared for a woman named Auguste Dieter in Germany for 5 years until her death, and then looked at her brain, she having presented with many of the symptoms that I described, this is what he saw. And what we see today at the gross level, compared to a normal brain, the affected brain has lost a lot of the volume of gray and white matter, the cellular material, the synapses, and so the solid material has shrunken, now leading to an increase of spaces between the brain occupied by fluid. And then grossly, the lesions which were identified by Professor Alzheimer, neurofibrillary tangles, which are intracellular, abnormal fibrils. The fibers that normally are the tracks that help to convey and transport nutrients through the cell. Abnormal phosphorylation of protein tau in particular leads to aggregation, disrupt cells and cell function. Extracellular deposits of senile plaques, which are composed of amyloid peptide and similarly have toxic effects on cells of the brain. These remained until recently, as I'll emphasize in a bit, the diagnostic criteria of Alzheimer's disease, which could be detected, in effect, only after death because one couldn't sample biopsy from the brain with any justifiability. But here's what's changed in the last few years that begins to change the story, and as you'll see, give a new kind of hope for making an impact in this disease, which has been resistant to date. So what has been developed are dyes that, when injected in a subject, a human subject as well as animal, will specifically bind to A beta or to tau. A beta is the material that makes up the plaques, tau, the material that makes up the tangles. This is a cognitively normal individual. For someone with dementia, if you use now a pseudo-color representation of the concentration of dye across the brain, you can see dramatic increases in both amyloid and tau in the brain of people who are affected. Now, just to point out that it's not such a simple story, there's a subset of people who are cognitively normal who also have some of these changes, and I'll show you in a moment why this is important and important to our understanding of the disease, how long it takes to progress, and what kind of opportunities there may be to be effective in changing the course of disease. So just this cartoon first to show you the formation of beta amyloid as we understand it. There is a normal transmembrane protein, the amyloid precursor, APP, that's this molecule. It is cleaved proteolytically by a series of enzymes, but in particular, the gamma secretases. There are two components encoded by genes called PS1 and PS2, with cleavage of this into a 40 or 42 amino acid peptide is the formation of a peptide which itself as a monomer or oligomer may be toxic, but which also forms the multimers that are the plaques that are visible. So there are at least 3 genes important here, one that encodes the APP itself, and the 2 that encode these 2 enzymes that process it. And I point that out because, importantly, mutations in any one of those 3 genes have been shown to produce an early onset, autosomal dominant, inheritable form of the disease. So discovered over the past years, beginning in the 1990s, were families around the world with a mutation in one of those 3 genes. If you have the mutation, with 100% certainty, you develop Alzheimer's disease, typically in the 40s, even 30s, much earlier than usual. And if a parent has kids, 50% chance they're going to inherit that gene. If they do, with 100% certainty, they'll develop dementia. If they don't inherit that allele, that mutation, they have the same chance as most of us of later in life having it. And I'll just take as an aside to mention one of the more moving or compelling meetings that I've been at over the last couple of years. Meetings on Alzheimer's disease have brought together families from around the world from these early onset families. And if you can imagine first just the impact it makes on these folks whose families are so affected to know that there are people around the world with the same disease and there's research happening that they can participate in. But picture young adults, your age or younger, folks in their 30s who've seen their parents die with Alzheimer's in their 40s and 50s, who have siblings who may have the disease, who may have young kids and have to figure out if they want to know whether those children have inherited the allele or not. So I point that out as background to allow us to understand what we can now do to track the disease early before there's symptoms. So in these familial onset cases, we can pretty well predict in each family when symptoms will appear. We can do brain imaging to look at amyloid and tau, as I mentioned. We can do that starting in 20s and 30s, and we can put that together in a composite to track how amyloid accumulates. Let's see if we can restart this for you. So we're here starting at minus 25. That's 25 years before onset of symptoms. And watch the clock tick, 22, 21, 20 years before symptoms, starting to see some differences already. Quite noticeable by 15 years before any symptoms appear. 10 years before, even more dramatic. And by the time 0, which is the time that the symptoms appear, extensive deposition of amyloid. You track the next several years with disease and progression, there's some change. But in fact, as I'll show you in a moment, most of the change in amyloid has occurred before the appearance of any symptoms. So this is a slide, yes? The question is whether the mutations are gain of function. They're dominant. So in that sense, it appears yes. They lead in general, so in the case of the mutations in amyloid, they lead to a protein itself, which is more susceptible to cleavage to give the amyloid the peptide that is susceptible to aggregation. In the case of the enzymes, in some cases there's quantitative, in other cases qualitative difference in their substrate enzymatic capacity, but by definition, gain in the sense it appears since they're autosomal dominant. We should say that in many of these cases, knowing exactly how those mutations, even though they've been well studied, lead to disease is far from clear. But what you can do is take any one of those human mutant genes, make it a transgene in a mouse. The mouse will acquire plaques in the brain. The mice do seem to develop more age-related decrease in cognition if you measure that in maze behavior, for example. And despite the fact we haven't yet succeeded in humans, there's been success in treating those mice by various strategies to reduce or reverse amyloid accumulation. It seems to reduce the amyloid and in some cases improve cognition. So mouse models can reproduce some of this and have some of the desired effects, which have led to a lot of attention trying to do this in humans, but as I'll point out, so far without success. So here is a series of markers. We're looking at time versus the biomarker abnormality. First, for the clinical symptoms themselves, this is either high risk or low risk people. This is developing from normal cognition to MCI, mild cognitive impairment, on to dementia. But if we take years before, look at amyloid PET, which is what I showed you, this red line, if we plot its accumulation in that graph, it is increasing, and if I had the scale plotted here, 10 and 20 years before appearance of symptoms, there's accumulation of amyloid. In fact, by the time symptoms have appeared, amyloid has long been at its maximum. There are other changes which appear, including loss of volume of the hippocampus, a part of the brain important for memory. All of these changes occur well before the onset of symptoms. So what does this mean in terms of what we can do? Until now, we've treated symptomatic disease and tried to slow its progression. That's really all we can do. But what this is illustrating is that have been treating at a time when many, many changes are far advanced. And now, for the first time, what we can do, and what studies have begun to do, in fact, is to treat prior to the onset of symptoms. So this is an example of one study that is currently ongoing. This is a family in the South American country of Colombia, which is the largest cohort of familial autosomal dominant disease. A group of people who are enormously committed and dedicated to research, obviously with great interest in making things better for their families and more globally. So if you look at people who are offspring who don't carry the gene, this is what they look like in their late 30s. In their late 20s, they're about the same. In the late 20s, the people with or without the gene don't look any different, but look what's happened to the gene carriers by the time they're in their late 30s. This is similar to what I showed you before. These are people who will develop dementia onset in their late 40s, and treatment has begun with therapies designed to decrease the accumulation of amyloid in people a decade or more before onset of symptoms, and hope that intervening this early will make a difference. We won't know for some time. First thing we'll do is track to see if it makes a difference in the accumulation of these biomarkers, including brain imaging. If it doesn't have any effect on the intended target, it's going to decrease the optimism for going on. If it does have an effect, if, for example, it decreases the accumulation of amyloid, that's going to be extremely encouraging for pursuing to see if it has an effect on the clinical outcome finally. And this is to show you the history of what genetics have told us about Alzheimer's disease overall. So back in the 1990s, I mentioned these three genes, APP, PS1, PS2 were discovered. There was relative inactivity between then and when the Human Genome Project was successful. We could map genes across the entire genome. So starting in the early 2000s out till now, here's an acceleration of many genes. And every time, now these are not causative. These don't account for disease with 100% certainty. They either increase or decrease the risk of developing disease by a statistically significant amount. Every time we find a gene of this sort, it provides a new clue for how we might design interventions targeted at those genes or pathways. And the genes on here are colored because also telling is the fact that they tend to fall into pathways. So the ones that are in a color code here, the darker blue, happen to all fall in the innate immune or inflammatory response system. And there are a number of these, and that's encouraged a good bit of interest in seeing whether interfering specifically with components of the inflammatory pathway and microglia in particular might be another strategy for intervening. So just some other fun facts, do with as you want. Relationship between sleep and amyloid. So you can decide which group you're in. 7 hours sleep or more a night, 6 to 7, less than 6. And again, by statistical analysis, there is more amyloid accumulation, age adjusted, more amyloid in those who get less than 6 hours sleep. Now, cause or effect? Maybe you sleep less well because you've got something going on, or maybe something about sleeping is protective. But I can tell you some very, very interesting recent laboratory experiments have shown in animals and some human studies that during sleep, a lymphatic or so-called glymphatic system in the brain that until recently was not even appreciated to exist is more effective in clearing, including the clearing of amyloid peptide, during sleep than it is when you're awake. So there's actually a biological credibility as well for sleep, and among the many things, we all try to imagine, what is it about sleep? We say it's restorative, we need it, but for what? This in fact may be one of those issues. So a good bit of research is now ongoing to try to understand cause-effect relationships of this using animal models as well as human studies. So let me turn then in the last minutes to talk about this relationship between biology of disease and biology of aging in a field that's been called geroscience. And geroscience basically is the hypothesis that basic aspects of age-related changes illustrated here, depending upon whether you read this Cell paper or this Journal of Gerontology paper, there may be 7, there may be 9. It's a bit arbitrary, but with a lot of overlap, there are common things which change. Macromolecular damage accumulates, response to a number of stresses, epigenetic changes, stem cells, metabolism, proteostasis, inflammation, most certainly mitochondrial function. Change, and the notion is that if we understand these changes and can do something to address any adverse effects they may have, this may have consequences on multiple diseases to which they relate. So I'll just end with a few examples of the kind of research that has been quite intriguing, suggesting that we have ways in which we can understand at a molecular level just what these changes are that happen with aging. Now, this is one approach to ask whether, if you compare a young and an old, in this case, mouse, we can identify factors in the young or old mouse which are responsible for aspects of its biology. And the technique that's illustrated here is parabiosis. I don't know how many of you are familiar with it, but it's what the cartoon shows. You take 2 mice in a rather limited surgical procedure, you stitch them together at the level of skin and underlying tissue, and they then go on to develop a common circulation. So circulating factors, and for that matter cells, are in common now between the 2 partners. And what's been called heterochronic parabiosis, which you can figure out just means heterochronic, different age, young and an old, put them together and see what happens to the young or to the old mouse in these combinations. And there have been some rather, let's see how we had this set up, some rather dramatic findings that respect to multiple tissues. One of the earliest findings had to do with muscle and repair of muscle from experimental injury. And what was found is when you connected an old mouse, which had slower muscle recovery from damage than a young mouse, and connected the 2, now the old mouse repaired muscle injury more like the young mouse. Something in circulation doing this. The heart is shown here because, again, in certain mouse models of aging, there is a hypertrophy of the heart that is associated with poorer cardiac function. And the picture here is meant to indicate that, once again, if you connect an older to a younger mouse, the older mouse with the enlarged heart actually reverts to a heart more typical of younger age. And shown around the center here are the fact that this affects a number of cell types. GDF11 is one of the growth factors that's been identified as playing a role here. So I mentioned this technique tells you something is in common in circulating, could be cellular, could be soluble. So people have done proteomic analysis and others comparing old and young serum, found candidates. One of them is a GDF11. Parts of this are controversial, part not. But you can see the pathway we're converging towards identifying molecules which may play a role in the function of multiple organs and tissues and may be relevant, therefore, to dysfunction or disease in multiple of those tissues. Okay. Epigenetic changes. So we're used to thinking the genome is what we're born with and we have it for life, and that's largely true, with the exception of mutations that occur at some low rate. But epigenetic changes are common with age and respond to environmental influences. And what a couple of groups have done is to plot, in this case, DNA methylation, but other aspects of epigenetic change as a function of age, and found that there is a very strong correlation of genome-wide methylation increase with age. Two groups have done it. This first slide is just to illustrate that two laboratories who did the same experiment found very concordant results. But interestingly, because at each age there's a variation, not everyone of a given age has exactly the same methylation. If you ask what the relationship is of methylation to aging in a relevant way, this is the lower and the higher quantile of methylation. So the group that has less methylation here and low methylation here, less methylation survive more with age, substantially. So survival is better in the group with less methylation. If you take that as a translation of genome-wide and perhaps organism-wide parameter of aging, gives a clues to the mechanisms by which aging may have an effect. And of course, one can affect methylation. That's not clear that doing that is the right thing to do, but it points out the very salient and central parameters of aging that do have relevance, not just because instead of looking at the calendar or your watch, you can tell time, but you can predict as a function of time whether someone is more or less likely to live a longer period of time based on these differences. Senescent cells. So senescence means a lot of things at levels of a lot of different disciplines, but it's come to characterize cellular senescence as a state in which cells have lost the ability to proliferate, essentially in response to any stimulus, and the usual definition, irreversibly. And it's become more apparent that those senescent cells are not simply passive, but also have acquired some active properties. So their secretory phenotype, including chemokines, cytokines, has changed too. So we have, with increased age, an increased number of senescent cells. How can you tell senescent cells? Well, they characteristically have changes in the expression of a number of molecules. One of them has to be P16, which many of you may be aware of, as relevant to cell cycle regulation, involved in tumor genesis as well. P16, highly expressing cells, accumulate with age. So what the investigators did here was to create a mouse first in which P16 drives a reporter, like GFP. So as mice age, you can look at various tissues, and the proportion of cells that are expressing high levels of P16 goes up. Now, it's still only a small percentage of cells, a minority of cells. The question was whether those cells were physiologically important or not. Now, if they were just passive, losing a couple of percent of functional cells when the rest were normal might not make a big difference. But if they're doing something active to interfere with the function of the cells around them, that's potentially more important. So they designed a way in which they could eliminate the senescent cells, the P16 high cells. So basically, one has P16 driving a genetic cascade, a transgene cascade, such that if you give drug, you will now suicide all the cells that are expressing high levels of P16. And you can tell that works because you can look at the mouse and see that those GFP positive cells are gone. And then you can ask what difference that makes to how the mouse works, how it functions. And this is just an example. I put some arrows here to show some of the differences. Looking at a couple of muscles, gastrocnemius and abdominal muscles, after deleting senescent cells, fiber diameter is increased with aging, so you're maintaining muscle, And if you look at their ability to work, for example, on treadmill, the time they walk before they exhaust, the distance they cover, and so the net work, they are capable of doing more work as a result of having reduced a population of senescent cells. So this genetic deletion of senescent cells is a little hard to envision directly translating to humans, but it's led to a series of screens for so-called senolytics, lysing senescent cells. People have screened small molecules and others and have reported some initial results in which these senolytics will do the same thing as can be done with genetic manipulations here, selectively removing these senescent cells. And they've begun to show, some of it just coming into press now, the same kind of positive effects on health by removing those cells with a senolytic drug. Well, you can imagine the interest, therefore, that arises in translating that into humans and how that will happen, how far that will happen, whether this is a good thing or a bad thing overall, hard to say, but just pointing out how complicated things can be in real life, senescent cells appear to have at least some beneficial effect. And it's illustrated here. In normal wound healing, over time, make a wound and then let healing occur. Normally, in this upper line, there's an increase in senescent cells during wound healing. It may just be that cells dividing, then getting to a point where they have to senesce, maybe even produce some senescence-associated factors to help wound healing may occur. You can eliminate those cells, as I mentioned, by this genetic trick. And down here, what happens? Well, it turns out that the wound size heals best in groups that form senescent cells. If you eliminate senescent cells, you don't do as well. So nothing is simple. So senescent cells may have some positive usefulness. It kind of makes sense that senescence as a mechanism wouldn't have evolved just to make you feel bad when you get old. But it's going to complicate, as all this does, the translation of this to real world. Genetics. So there are endless studies, I'm not going to show you any of them, which in yeast and worms and flies have shown that simple single gene manipulations, mutations, can double, triple, in combination up to six-fold increased lifespan. That's led to a lot of speculation by some that we ought to be able to do the same thing in humans and live essentially forever, and you may have read some of those articles. Again, the translation there is not so simple. But here is one of the few cases in humans about what's known about genetic polymorphisms that are associated with age and how long one lives. And this is FOXO3, for example, which does make a difference. Now, what FOXO3 does, how it does it, whether we can translate that information into doing something good for people so they live long and live healthy, we don't know. So the summary of these few dimensions I've talked about with you, the world's growing older. Sadly, life expectancy is actually decreasing for some groups. Not everyone is having the advantage of biomedical and public health changes, and we have to do something about that. Age is a risk factor for a whole lot of diseases. We've learned a lot about some of them and made some very important differences. And now, Geroscience, if there's a new perspective, is asking whether, as we learn about basic underlying processes of aging that contribute potentially to many disorders of aging, decreases, functional decreases that accompany aging, we can use that information to make a difference, not just for a single disease at a time, perhaps have a global and positive impact. So I thank you for the time and patience and leave you there.
Thank you very much, Richard. That was spectacular. So we have time for some questions. Try and use the microphone.
Could you elaborate on the relation between neurofibrillary tangles and amyloid deposits? Because there was a paper recently in Science that said specific phosphorylation of tau on position 38 reduced tangles and reduced deposits, although I think it was in a mouse.
Yeah, so this is, plaques and tangles, as I showed you from the beginning, from 1906, have been the sentinel changes. And in recent research history, there's been a great deal of discussion and even controversy about the relative importance of the two. So beta amyloid plaques, BAP, has been the favorite of so-called Baptists, and tau for Taoists, if you take the play on two religions, have characterized the groups which somehow by belief will favor the predominance of one or the other causally. If you track what happens with progression in humans, now that we can do that, it's interesting to see what occurs. Amyloid occurs first, and you accumulate amyloid. It appears that when the amyloid load reaches a certain point, then tangles, which initially appear in just an isolated area of the brain, begin to spread. And the spread itself is interesting. It appears to be a transcellular spread of tau abnormalities that is moving from cell to cell. So in the end, I think people would generally say that cognitive change now correlates better with tau than amyloid, but whether the tau changes are triggered by amyloid, and both are important for the ultimate pathology, remains a subject of a very important conversation, if that's clear.
Does progeria contribute anything to our knowledge of aging?
Yes, so progeria as a general term, it really refers to premature aging. And perhaps the condition that's been associated with it is Hutchinson-Gilford, a mutation in a lamin, which leads to abnormal splicing. I mean, among the people who have studied this extensively has been Francis Collins, and it produces abnormalities in nuclear membrane. Now, some of these changes also happen with aging in senescent cells. The kids who have progeria have a lot of the characteristics of premature aging, hence the connection. And so as we're learning or trying to learn the way in which we can treat the kids with a genetic disease, the question in parallel arises about whether these same treatments or interventions will also affect the changes, similar changes that occur with normal aging.
If I understand it correctly, in one of your graphs, looks like the FDG PET was increasing after the beta amyloid increases. Does that mean glycolysis was increasing, and why would glycolysis increase there?
So FDG PET is generally regarded as a reflection of metabolism and maybe therefore related even to synaptic activity in the brain. So as a surrogate, albeit indirect one, for the number of brain cells, the number of synapses, and the metabolic activity of the cells. So the fact that it's occurring a little later means that it's somewhere downstream, for example, of the accumulation of amyloid, before you actually start to see neuronal cell deaths and synaptic dysfunction, which is all reflected in decreased metabolism by FDG PET. Decreased. Yes, I'm sorry. What was the way, yeah, if the slide was misleading, it was showing the increase in abnormality. In some of those cases, and I've got a better one that shows some up and some down, but it's just meant to show you the changes occur early. That's right. So if hippocampal volume had been posted there the same way, it would look like it was going down. So it's percent abnormality, whether that be up or down. Sorry for that confusion.
So since you mentioned chromosomes and senescence, also what about telomeres and telomerase activity in senescent cells? And as we know, like lobsters don't have it, right? They can live forever. So how do they do?
So telomeres, just to catch everyone up, telomeres are the structures at the end of chromosomes. They are in mammalian cells. They're multiple repeats, TTA, GGG repeats, for example, because of the requirement for template in RNA replication during mitosis. There is imperfect elongation of the telomeres at the ends. Template sits there to initiate. You don't reproduce the portion where the template sits. So in theory, you lose nucleotides. Estimates experimentally been maybe 50 to 100 nucleotides with every cell division. Telomeres get shorter and shorter. If the telomeres get too short, they actually lose their T-loop configuration. They no longer are protected. They look like DNA breaks. There is a response to as if to DNA damage, and cells become dysfunctional. So having said all that, you can show very well in, well, for example, a mouse model. In fact, my own lab and others have made mice that are deficient in telomerase. Telomerase is an enzyme, which you can tell by the name, it adds telomeric repeats. So it compensates for the fact that you're losing telomeres with cell division. Telomerase extends them again. Knock out telomerase, You don't repair, and every generation of mouse, the telomeres get shorter until you get to a point where some of the characteristics of aging happen. Among others, they become sterile, they're infertile, they can't reproduce anymore. You can repair that by putting telomerase back in. That's in a mouse. In humans, if you plot with age, telomere length versus age, telomeres decrease with age. There are specific diseases of essentially telomerase dysfunction in humans which cause disease because of short telomeres. In fact, there's what's called anticipation, meaning in successive generations of this inherited disease, the condition gets worse because in the germline, telomeres are getting shorter. So each generation has less tolerance for telomere shortening. So this is long about to get to your the answer to your question. There are diseases in humans as well as animal systems where shortening of telomeres leads to serious consequences, even death. If you ask the question, though, in normal humans, in absence of these specific genetic syndromes, what is the evidence that telomere shortening plays a role in senescent cells or in health or longevity? It's far less clear. There are many reported correlations between telomere length and even life expectancy, but which is cause and which is effect, not yet clear. And there are people who are talking about interventions to elongate telomeres, stimulate telomerase in order to treat and provide health. Some immediate cases where it can be applied right away, if you think this is an age when we're all very interested in immune therapy, for example, for cancer. Take cells from an individual, activate them in a way that they now are anti-tumor cells, put them back in. If those cells, typically T cells, have short telomeres, they won't live very long in the person you put them back into. They'll disappear. If you can elongate their telomeres, they may live longer and work more. And so that's more immediate cell transfer, regenerative medicine kind of approach. But whether in the whole human, aging can be impacted by affecting telomere shortening, not clear yet.
Well, thank you very much, Richard. That was marvelous. And I think we should have every time for some questions afterwards after Toren.
Thank you very much. It's a pleasure. Can you hear me? Following Richard and our patient. It's good to know. I was impressed that if you got less than 6 hours of sleep, you're at risk for getting dementia. So I often look out when I'm talking, I see people nodding off and dozing, and I've always interpreted that as I'm being boring. But now I know they're just protecting themselves from dementia. so go to sleep, ask questions, do whatever you wish. But I'm going to talk a little bit about aging and why I think it's so interesting biologically. I think all of us can recognize what aging looks like. As we said, it doesn't take an Einstein to figure out what old and young look like. But as Richard mentioned, the underlying mechanisms of aging have been really relatively elusive. And in fact, I think, for me at least, aging represents probably the most important but least understood biological process out there. And I think one of the interesting things and why I like talking to audiences like this is to get people more interested and excited about it, because I think it is so fundamental and so fundamentally poorly understood that we need more people interested in this very, I think, very important question. So why is it important to understand the basis of aging? So as Wynn mentioned, I was trained as a cardiologist, and as Richard mentioned, cardiovascular disease is the most number one killer of people in the U.S. and the Western world. Heart disease kills more people than any other condition. But if I were to say the National Heart, Lung, and Blood Institute, which I am a member of, and research finally paid off, and we were actually able to cure all cardiovascular disease, so that someone like myself was now immune from getting a heart attack or a stroke or heart failure or any of the other cardiovascular conditions. And therefore, we've eliminated the number one cause of death in the U.S. How much longer do you think, there's not a quiz here, but how much longer do you think I would live because of that great research breakthrough? Anybody want to give an idea? 5? Any other ideas? 10? Do I hear? I feel like an auctioneer at Sotheby's. Well, the answer is about 3.5 years, which is not bad, but not that much if you think about it. And if We took all the money from the Heart, Lung, and Blood Institute, which I'm not recommending, and now gave it to the NCI, which is the second leading cause of death, and eliminated all solid tumors and hematological tumors in people, we'd gain another maybe 3.5 years. So getting rid of all cancer and all heart disease would only extend the lifespan of someone like me about 7 or 8 years. Again, not terrible, but not as much as you would think. And why is that? And as alluded to before, because when we get older, a lot of bad things happen, unfortunately. When I got interested in aging, I was about here on these curves. And as you get further and further to the right, they become more and more personal, I can assure you. And so here's the incidence for Alzheimer's disease and cardiovascular disease and cancer in the US population. They all look the same, and enriched to other curves of other diseases. That is, are very rare as we're young, and they exponentially rise as we get older. And so one of the tenets, I think, of aging, and one of the promises is that, most of the NIH is devoted to thinking about curing these diseases by focusing specifically on the y-axis. That is, coming up with drugs or treatment strategies that will affect cardiovascular disease specifically, or Alzheimer's disease specifically. What I'd like you to think about today is not the y-axis, but think about the x-axis, right? And that is the promise, I think, of aging research, is that we don't worry about the specific causes per se, but try to move things along the x-axis, try to extend lifespan, try to push all these curves to the right by slowing down the fundamental process of aging itself. So how far can we push these curves to the right? How long can we live? So the answer is probably shown here. And this is Madame Jeanne Calment on her 121st birthday. She is, if you exclude Moses and Methuselah, the oldest recorded lifespan in history. And this is her, as I said, on her 121st birthday. She lived almost another 2 years, to almost 123. She had a very nice life, as far as I can tell. I never met her, but she lived in southern France all her life. She lived in Arles, which, if any of you have been to, is spectacularly nice. Her uncle owned, so she lived, during the entire period of the French Impressionist movement, and Arles was sort of the center of much of that. And her uncle owned, it turns out, an art supply company. And so a lot of the French Impressionist painters would come in and buy supplies. She had very nice things to say about Cézanne, who she said was lovely. And she said Van Gogh was very disagreeable, as you might expect. So my favorite story about her is that, I don't know, this custom doesn't exist in the US, but in France, I guess, if you have a very nice apartment, people can pay you a monthly retainer with the idea that when you pass away, they will get to basically have your apartment. So when she was in her late 80s, a very nice gentleman down the block who coveted her apartment, who was in his 40s, began paying her this monthly supplement with the idea that he would inherit her apartment. And as you can imagine, 30 years later, when he was in his 70s and she was in about 110, he passed away and she remained in that apartment. And even though no one else took her up on the challenge, even though she was of 110 to pay her monthly. So she actually lived there to about 118. There's another story about why she had to move, but I won't go into that. So did she do something, like as our patient was talking about, did she do something that was extraordinary that explained her lifespan? And the answer is no. She smoked. This is her smoking. She smoked to 119. She drank every day. She did all the things that we're not supposed to do. As mentioned before, it's a little hard to gain that much inference from people like this. In fact, let me ask you another set of questions. So, as I said, how many people here believe that genetics is the most important determinant of their lifespan? That their genes that they inherited is what determines their lifespan? How many people think it's the environment? Well, obviously it's both, but most important. Yeah, so what?
Depends how bad it is.
Yes, that's true. If you live next to a Fukushima reactor, right, it doesn't matter how good your genes are. Right. So it's probably not a simple answer, and there are probably, especially for people who live extremely long lives. But for most of us, the answer is that genetics account for about 25% of our lifespan. The most of it is environmental. So for instance, her children died in their 40s, one from a car accident, and one got pneumonia in the days before antibiotics. So lifespan is complicated, and the genetics are complicated. But clearly, genetics has a role, as does environment. So what are the environmental influences that are important? And so one of the things that's been very interesting for people like myself to try to analyze is this idea of caloric restriction, or the amount of calories we eat as a determinant of our lifespan. And it's been observed and known for about 100 years now that if you take laboratory animals and feed them less, they tend to live longer. And that's a very robust phenomena. It's robust in the sense that the effects are quite large in animals such as mice and rats, and robust in the sense that it's pretty well evolutionarily conserved. So you can see similar effects in yeast if you under certain experimental paradigms. And as I can say, that response is conserved all the way up to mammals, certainly at least rodents. The data in higher mammals like primates and us are a little more controversial. But nonetheless, people have been very interested in trying to understand what is it about eating less makes you live longer. And I won't go through all the details, but just to say that there are two sort of pathways that my lab has been interested in that seem to sort of mediate, at least partially, these effects. One of them are these families of enzymes that I won't talk about today, which are the sirtuin family, which are NAD-dependent deacetylases. And the other one is this protein mTOR, which I will talk a little bit about, and just go over a mouse model. Again, not because it's incredibly interesting, but just sort of gives you a sense of what can be done and understood about aging in experimental paradigms. So what is mTOR? Many of you know it's a large protein, the serine threonine kinase, that acts inside the cell as sort of an energy sensor. So it senses the availability of a growth factor stimulation or nutrients. And when those factors are present and abundant, mTOR is activated, and it acts to then allow the cell to grow and expand and get bigger by regulating a variety of processes, including protein synthesis and metabolism, as well as inhibiting a process which we'll come back to, which is autophagy. In contrast, when nutrients are low, mTOR is inhibited, and the reciprocal events happen in these processes downstream. And so in lower organisms, it's been shown that reducing mTOR activity genetically produces an increase in lifespan. And the National Institutes of Aging has this very interesting program called the Interventional Testing Program, where they test different molecules every year, usually about 5 molecules a year, to see whether or not they can prolong the lifespan of mice. And several years back, they got a very nice hit, as they would say, by giving mice this drug rapamycin, which many of you know is an mTOR inhibitor. They had a little trouble, even though the molecule was accepted into the program, they had a little trouble formulating the rapamycin for the mice. So they only began feeding the mice rapamycin roughly when they got to be about middle age. But nonetheless, even giving the mice rapamycin from middle age on produced about a 10% increase in lifespan for the animals. So again, here's this idea that maybe that aging, which we've always accepted as being a risk factor disease, but we've always thought of as being unmodifiable, now begins to feel that you can begin to say, well, maybe aging is not unmodifiable, and that you can manipulate it by the small molecules such as rapamycin and sort of extend lifespan, and therefore can you then begin to think that you'd have effects on diseases. So we're interested in this phenomena that inhibiting mTOR might increase lifespan. So we wanted to provide a genetic example of this. And that's a little difficult with mTOR. So as I mentioned, mTOR is a very important protein, and it appears from both the rapamycin studies in mice and studies in lower organisms that if you reduce mTOR activity, that animals live longer. So the prediction would be that if you got rid of mTOR, you would live forever, but that's not the case. If you get rid of mTOR in mice, they die in embryos. So the secret in all these things is that you can't get completely rid of it, but somehow reducing its activity is beneficial. And so I won't go through the detail. mTOR comes in sort of 2 different protein complexes that are distinguishable by the differences in associated proteins. But basically, if you delete mTOR or any of these associated proteins, all of them are embryonically lethal. But fortunately, Beverly Mock, who's a colleague of ours here at NCI, had, for reasons that I won't go into, generated a mouse in which there was a neomycin cassette inserted in the mTOR locus. And so this neomycin cassette was inserted in the intron of the gene. So it doesn't affect the coding sequence of the gene. But it did affect the transcription and translation of this mTOR allele. So we called this the delta allele because the expression of this delta allele was the normal mTOR, but was expressed at much lower amounts. So if you, so this is just an example of a Western blot of mTOR expression in wild-type mice. These are cells from wild-type mice, or cells derived from what we call these delta-delta or hypermorphic mTOR mice. And this is from cells, but we did it from tissues too. And we found that this mTOR was the normal mTOR, but it was expressed at about 25% of the normal level. Okay. And so this work was done by Julie Wu, who was a postdoc in my lab, who's now left the lab. So what is the effect of reducing mTOR expression by about 75%? So again, we're not getting rid of the gene. This is one of 30 or so thousand genes in the mice, right? We're just changing the expression of this one gene by about, and reducing its expression by about 25%. Oh, sorry. So so these are the mice. They're born. They're a little smaller than the wild-type mice, and that weight persists. I should say, when this paper came out, the Wall Street Journal did a little story on it, and they wanted a picture of the mice. They normally draw things by hand, but they wanted actual pictures. It's very hard to get mice to pose. I don't know if you've had this problem. And they were a little nervous, so this made it into the Wall Street Journal, but this mice peed here. I don't know if you can tell that. It was this big moment, and he was nervous. What can I say? Okay, so these mice are a little smaller, but otherwise look very healthy. Okay, I won't go through this because we're a little late. And this is the lifespan. So after 3 years, Julie found that these mTOR hypermorphic mice, either the males or the females or the whole group, live longer. Okay, so again, we're changing one gene. We're reducing one gene out of 30,000. And the lifespan extension for reducing mTOR in these mice was quite significant. They lived about 20% longer. Okay, so that's the equivalent for us, if you can make the equivalence, of going from a mean lifespan of about 75 years to a lifespan of 90 years just by altering a single gene in the genome and not even getting rid of it, just by reducing its level and activity, which is remarkable. Okay, so again, we're supposed to end at 5:30, right? So I'll speed up. I'll speed up. So these mice live longer. So one of the things we wanted to ask, what? We can go to 6. Okay, we're all aging together then. So these mice live longer. So one of the things we wanted to ask is, does an intervention that makes you live longer slow the aging in your tissues? And if so, is that slowing uniform or not? That is, what is the relationship between our lifespan and the aging in our tissues, right? So we said, okay, let's phenotype these mice and look for the age-dependent decline in organ function in control mice or mice that we know are going to live longer, okay? So we did this. We did a lot of these tests, okay? So I don't want to go through all of them. But here's an example of, this is a test of memory and learning in the mice, okay? So, and again, this is an example where you take the mice and you put them on a raised platform, and in one of the there's a bunch of holes around this platform, and in one of the holes is an escape for the mice. And the mice don't like to be up high. It's very bright. They don't like to be bright. So they're very motivated to find a way out of this situation. As I say, they don't know this has been approved by the NHLBI Animal Care and Use Committee, and that nothing bad could ever happen to them. They're very motivated because they haven't read the protocol. So in one place, there's an escape. And so you train them. And in the beginning, they're just frantically looking for it. But then they realize that there's this hole, the escape hatch. And after a few days, they get less nervous, and they basically beat a line to this to where they should go to escape. And so you can do a number of tests to show that they're actually focused and directed there, and you can time it how long it takes them after 3 or 4 days of training to find the right place to go. So again, in the beginning, when they're young, they're very good at this task. And there's no difference between the wild type and these long-lived mTOR hypermorphic mice. So mice get older, they're not as good at this task. They don't remember as well, they don't learn as quickly as all of us. Like, I don't remember where I parked my car today, I'm a little nervous, I'm not gonna find my way home. But, so the mice are the same way. It takes them much longer, they're not as good at memory and learning. And you can appreciate, this is the control mice at sort of an old age, and these are the hypermorphic mice. They're not like young mice, but they're better than the control mice. And we could do a bunch of tests to show that the capacity to learn new things was much more youthful, right? So that would suggest, like they live longer, their age-dependent decline in memory and learning was also better. And I don't want to go through all the details, but here's some other tests we did. Here's a test of their ability to stay on a rotor rod, which is a complex thing of sort of muscle function and neurological function. But again, you can appreciate that in this case, as the mice get older, they're less good at this task. But again, these long-lived hypermorphic mice are somewhere between the young mice and the old mice, that their function is preserved. Here's another example. This is measuring stride width variation in gait, which is actually a very good predictor in elderly people, elderly humans, of who will fall and who won't fall. Mice are on 4 legs, so they never fall. But again, their ability to maintain normal stride variation is impaired as they get older. And again, the hypermorphic mice are acting more youthful. So we did Julie did a bunch of these tests. And in the vast majority of them, the mice that live longer by manipulating mTOR look like they had improved their function, organ function. But there were exceptions. So here's an example of bone density. And again, as these mice got older, bone density declined in the control mice, but they declined even more in the hypermorphic mice. And here's an example of superficial infections that the mice get as a function of age. So again, these are the control mice, and these are the hypermorphic mice that live longer, but got more of these infectious problems than the control mice. So what is the conclusion from all this? I don't know. I don't know. But I think there's 2 things that we took away. First is, again, this is, I think, again, the power of genetics and the power of, I think, of aging biology, that on the one hand, you can make single genetic changes that have profound effects on lifespan. And I think this is one example, but there are many other examples now in both mice and other organisms. But the second thing is, when you start doing in-depth phenotyping of these animals, which you can't do in lower organisms, like you can't ask flies and worms to do much, and even mice, it's difficult in many cases to figure out what's going on. But when you start phenotyping mice in depth and looking at the aging of organs as opposed to the organism, that you find that manipulations that extend lifespan do not affect each organ similarly. And it suggests that aging, which is complex already, is probably not the same thing, and that each organ may age at different rates or under different mechanisms. And I think that's something that clinically people are very well aware of, that you have people who get older who have terrible blood vessels but very good bones, and vice versa, right? So that there is something that the aging is not uniform, and probably this suggests that it's not mechanistically uniform as well. Okay, so I'm going to. How long do you think I should keep on going? What do people say? Do you want to hear the short or the long version? Okay, all right. I got nothing to do. Okay, so how does mTOR work? So we don't know. And as I said before, there's a bunch of things that it controls, and it's very difficult to sort out at this point why reducing mTOR makes you live longer. What are these downstream effectors that are really the most important for driving the phenotype? But I can say what we're the most interested in. We're the most interested in this process called autophagy, and whether or not it could be an important regulator of aging. So I just wanted to spend a little brief time on how trying to look at autophagy and genetic models that we've created to try to understand the relationship between aging and autophagy, as well as other genetic models that allow us to look at rates of autophagy in vivo, which we think is an important way of thing to do. Okay. So what is autophagy? So as many of you know, it won the Nobel Prize this year, and it is the process first described in yeast, but very well evolutionarily conserved, by which damaged proteins and particularly damaged organelles are incorporated or encapsulated in a membrane, a double membrane structure that is formed de novo in the cell that engulf these damaged cargo in what's known as the autophagosome, which can then fuse to the lysosome and deliver those damaged cargo for degradation. So there is a variety of evidence to suggest that autophagic flux declines as we get older. And I think you can appreciate how that might provide a lot of problems for us, right? So if autophagy declines, and autophagy is the way that you get rid of damaged proteins and damaged organelles, in the setting of a decline in autophagic flux, you're going to start accumulating garbage, basically. It's as if, you know, if the Montgomery County didn't come and take your recycling bin every Thursday, in my case, probably different days in your case, but eventually the trash builds up, right? And in your cell, if you think of autophagy as a recycling system, then you can imagine that as you age, if this process slows down for whatever reason, that the trash will start building up. And what is that trash? That trash is damaged proteins and damaged organelles that normally would have been removed. And we're particularly interested in this accumulation of damaged mitochondria because we showed previously that, and we've always been interested in this, the relationship between aging and oxidative stress. And certainly in the absence of autophagy, there is a buildup of mitochondria that are normally removed that contribute to this high rates, we think, of oxidative stress in elderly tissue. So one way we began to approach this is to try to say, does a defect in autophagy recapitulate aging to any degree? And since we're in the Heart, Lung, and Blood Institute, the models that we started to look at this in is in the blood vessel. And so the question is, there are a number of changes that occur in the blood vessels as we age that contribute to vascular disease. And to what degree can we mimic or phenocopy these by disrupting autophagy within the vessel wall? And we're just beginning these studies, but we have some preliminary results I want to show. So here's one example where we've knocked out autophagy in the endothelium of a mouse and fed those mice high fat and asked what happens to atherosclerosis. And so here's an example of the control artery, aorta, and here's an example in which we've disrupted autophagy only in the endothelium, and you can see that there's significantly more atherosclerosis that's formed in these versus the control animals. Here's another example which we're still working on, Yi Yang in my lab is working on. In this case, we've disrupted autophagy in the smooth muscle layer of the vessel wall. And this was alluded to before. This is an example. This is work from another lab. This is actually work from Francis Collins' lab in a paper published in 2006, in which they have a mouse model of this syndrome that was talked about briefly before, Hutchinson-Gilford progeria. And those mice and those people suffer from cardiovascular, the major cause of death is cardiovascular disease. And if you look in their blood vessels, in the aorta, in the mouse model and humans, there's a loss of smooth muscle cells in the aorta that occurs over time, and a change in what's called the internal elastic lamina, as opposed to the control mice, where you see these are all sort of nice and wavy. There's straightening and breakage of the internal elastic lamina with a loss of smooth muscle cells. And what Yi has found in our mice, where we've disrupted autophagy in the smooth muscle cell layer, is a very similar phenotype. So that a defect in autophagy in the smooth muscle cell in many ways is phenocopying this accelerated aging phenomena. So again, this link, we think, between perhaps a decline in autophagy and the phenotype of aging. So that's something we're obviously very interested in pursuing. Okay, so let me just end by talking about ways that we can begin to measure these processes in vivo. So as I said before, we're very interested in mitochondrial turnover, this process of mitophagy, that is how damaged mitochondria get removed through the lysosome and autophagy. And so what we've done is to develop an in vivo assay to begin to measure this. This is work done by Nuo-sun in the lab. And it's really based upon this assay that a group in Japan first described by this protein KIMA, which is a coral protein, which has two nice properties that make it useful for this assay. So when we direct KIMA to the mitochondria, we call it mitokima. The properties of KIMA that are important here is that its excitation is pH dependent. So it can be excited when it's in the mitochondria. The mitochondria are actually the most alkaline organelle in the cell, so they have a pH of about 8. And KIMA will be excited in the green wavelength under those conditions. And when KIMA is delivered to the lysosome, it's in a very acidic environment. The lysosome is the most acidic organelle. And KIMA can then be excited in the red wavelength. So that's one property of KIMA that's very important, that its excitation is pH dependent. The other property is that although it's a protein, it's very resistant to lysosomal degradation. So it gets delivered to the lysosome, but it stays in the lysosome. And so one can image with a green excitation and a red excitation and generate an image like this from a cell and get a sense of KIMA that's either in mitochondria or KIMA that's been delivered to the lysosome. And this green to red fluorescence is therefore a measure of mitophagy. So let me I love movies. I never have movies in my talk, but I finally got a movie that I can show, so I got to show it. So this is cells that express KIMA. They also express this protein parkin, which, and these are cells actually Richard Yule gave us, who has described the role of parkin in this process of mitophagy. And so in this top row, these are KIMA cells and parkin cells where we damage the mitochondria to cause mitochondrial damage with these pharmacological inhibitors. And in these bottom row, we're just imaging these cells without mitochondrial damage. And what you'll see is that these cells that get damaged will recruit parkin to the mitochondria. So this parkin, which is sort of homogeneous in the cell, will rapidly go to the mitochondria, and you'll see these punctates. And that will start this process of mitophagy, which will change this green fluorescence to red fluorescence. That's a lot to say. Okay, let me just start it. Okay, I'll just stop it there. But now you can see that as opposed to how it was here, that parkin is now recruited to these punctate structures, which we know are mitochondria. And you're beginning to see these red dots appear, which represent mitochondria that have been delivered to the lysosome. And this KIMA protein has now been able to be detected in the red wavelength. And if we let this go over 18 hours, in the control cells, which we haven't damaged, there's a relatively low rate of mitophagy. In contrast, in the cells which we've damaged the mitochondria, all the parkin basically is gone because it's been delivered to the lysosome and it's degraded there. The KIMA has also been delivered to the lysosome, but it's relatively stable, so that you see this persistent red fluorescence, okay? So basically all the mitochondria have been turned over in this very harsh mitochondrial damaging condition. So this is in vitro. So Nuo-sun in my lab created a mouse that expressed this in all the tissues, and that we thought would give us a way to look at mitophagy in vivo for the first time. And we did a number of things to show that this model is correct. I won't go through it, but in general, what we saw is that there are very large differences in mitophagic flux in different tissues. There's also incredible differences within tissues. So here's an example, though, of the brain. And interesting, this is the hippocampus. This is another region in the brain where there's high levels of stem cells. And in those regions, there's high levels of mitophagy compared to other regions of the brain where that doesn't exist. And this is sort of the reason why we perform the experiment. So this is again looking in the area of the hippocampus in either young mice or old mice expressing this KIMA reporter. And in the young mice, this area of the hippocampus, this red or orange color represents high rates of mitochondrial turnover. In the same region of mice that are old now, the mitochondria are still there, but there's relatively little turnover, as evidenced by the absence of the orange and red color. So this provides, I think, an example where there's clear evidence that rates of autophagy, or in this case, mitophagy, decline as animals age. Remember before I told you about this memory and learning test? This ability to remember and learn is all encoded in this region of the hippocampus. And so as we get older, and mice get older, as I mentioned, your ability to remember and learn goes down. And at the same time, what we're showing here is that this rates of mitophagy goes down as well. And so we're very interested in understanding, does in fact this decline in mitophagy and autophagy contribute to this problem of remembering and learning? And one way to go about that is to develop drugs that stimulate this process. And so Nuo and others, with the help of the NCATS facility, have used the same chema-based assay to ask, can we find small molecules that stimulate mitophagy? And so we're just starting this process, but we've screened 3,000 small molecules that will stimulate this chema-based assay in cells. And we got about 20 or so of these compounds that look like promising compounds. And just as a readout, and again, this is very preliminary, we took some of these compounds and gave them to flies that had a model of Parkinson's disease. And at least in the first go round, it appears that some of these compounds may be efficacious in treating that condition, suggesting that if you can increase mitochondrial turnover, you may have beneficial effects in Parkinson's disease, and we would argue a variety of other conditions in which mitophagy slows down and may contribute to the pathology. So let me just end there by saying that I think this is a phenomenal time to be interested in aging. I hope some of you are swayed by Richard's talk and my talk to throw away what you're doing now and get on the aging bandwagon. And the hope is that, as I said, if we can move the x-axis, that is, if we can treat the underlying fundamental mechanisms by which we age, that we might be able to develop a way to treat a variety of diseases. And I would argue, since we're talking about X and Y, that it is truly orthogonal, that is, to the way that we treat diseases now. And I think that's the hope and promise of why, and the excitement of why studying aging is so fundamentally interesting. Let me just end by just thanking a few people who did the work. As I mentioned, Julie Wu did the work on mTOR. She's left the lab. She now works at the patent office here in town. Yi Yan is doing this work on autophagy and the vascular wall. And Nuo, as I said, is doing our work on mitophagy. Thank you, and I'd be happy to answer any questions.
That was great, Toren. So we have, would you pass it?
You can just shout.
Why do you need high mitochondrial flux? Why are they particularly more defective than other intracellular organelles? I mean, why don't they just kind of repair their little bits that go wrong and keep on going?
Right. So there is some evidence that and I don't know if it's been published yet, but there is some evidence that mitophagy does not have to involve the entire organelle, that you can actually butt off part of what's the quote damage about the mitochondria and leave the remaining part of the mitochondria there. But I think the better answer to your question is that mitochondria don't have the sort of DNA repair machinery that the nucleus have. They have their own genome, but they don't have the DNA repair. They're also the site, obviously, of metabolism, which is an oxygen-independent ROS-generating phenomena. So they are, by estimates, produce about 90% of the reactive oxygen species within the cell, and their DNA, which is sitting there, doesn't have the intrinsic DNA repair mechanisms that nuclear DNA system has. So they're at the site of, basically, they're the epicenter, the ground zero for ROS production without sort of the repair mechanism that the nucleus has. So there has to be a way to sort of, people think of regenerating them.
I wonder if the drugs that you use for reconstituted mitophagy that you showed towards the end of your talk, do they have any feedback effect on mTOR activity?
So we don't know yet. I mean, we're just playing around with them yet now, and so we don't know. But that's certainly a potential.
So I know that rapamycin has some unfortunate side effects. But are there any studies looking at low doses of rapamycin in aging in humans?
Yeah, so some of the side effects in mice are testicular atrophy. So I think you're right about half the population signing up for rapamycin therapy. So I think the good news about rapamycin is, as I mentioned in the first study, they gave it in the mice basically from half the lifespan on. So there have been now in mice studies of using lower doses and intermittent rapamycin, and they can get many of the same effects by using those types of strategies. And there, so the data on rapamycin and lifespan have not been done. There has been one small study using a rapamycin analog in people that was sponsored by Novartis in one of their rapamycin inhibitors, mtor inhibitors, where they took, and this is sort of interesting because rapamycin was developed as an immune suppressant, right? But as many of you know, as we age, our ability to generate an immune response goes down in what's called immunosenescence. And so they took people over 65 and gave them this mTOR inhibitor, this rapamycin analog, and asked whether or not it actually improved their immunological phenotype. And in fact, when they get, and so the study, the intervention was a flu vaccine, and many elderly people cannot respond well to a flu vaccine. And in this small study, it was shown that people who got the rapamycin inhibitor actually did mount a better immune response than the controls, which is counterintuitive if you know, so I mean, it's small, it's one study, it's one aging phenotype, but it is at least suggestive that maybe things along this will work.
I was just wondering if you'd looked at metabolism between your old and young mice and whether there was actually any significant differences in OXPHOS or anything like that.
So in the mTOR mice, so right, so we had made some connections between mTOR and mitochondrial metabolism, but as far as we could tell in these mice, the mice are smaller, so at least in total body oxygen consumption, when you normalize for body weight, and people differ on how they do that, but the way we did it, we could see no difference in metabolism between the wild type and the hypermorph. So we don't think that there's a clear metabolic basis for the differences in lifespan.
So, Toren, do you think there's a connection between the studies and the parabiotic mice
and
TG? What was the name of the
GDF11?
GDF11.
Yeah, so that's thought to be work in the TGF beta. Is a TGF beta sort of molecule. I don't know what connection that has, if any, to mTOR activity or to mitophagy. We actually tried to see if GDF11 stimulated mitophagy, but we didn't see any effects. So.
Has anyone studied mitophagy in plants at all?
I'm sure they have, but I don't know. I don't know. I mean, I don't know if I don't know what the equivalent chloroplasty or chloropuff. I don't know, but I don't know.
Mitochondria as well.
Yeah, no, I do. Yeah, absolutely. But I don't know to what degree it's been studied.
Do you want to say a comment about mitochondrial dynamics in, say, in tissue culture cells as compared to in organs or in organs, the fission fusion?
Yeah, I mean, I think you know, I think fission and fusion. So fission and fusion are related to mitophagy. They're not, you know, obviously the same thing. But a lot of people think that, you know, that you need fission for mitophagy to occur. And the rates of fusion and fission in vitro are much higher than they are in certain tissues like the heart. You basically don't see fusion. You don't see fusion and fission with any sort of, you know, regularity. So I think there's a lot in cancer cells in culture. You see a lot more of these phenomena than you do in vivo.
Do you think the quote aging phenomena in different tissues is related to the different morphology of mitochondria in different tissues?
I don't know. I mean, I don't know to what degree, you know, in that case, we're making a genetic mutation in mTOR, and I think the simplest explanation is that the role of mTOR varies between different tissues in terms of its role in aging.
Thank you. Could you pull out the slide with mTOR diagram?
the cartoon of what mTOR does?
Yes, and so there is mitophagy that goes down, and there is when all these factors and nutrients are abundant, and metabolism goes up.
Well,
So yeah, so the question comes that like in young growing organism, what do you think that the protein synthesis goes up, autophagy goes a little bit down? So why there is decreased lifespan?
no, so when mTOR goes down, protein synthesis goes down. So people think that it's not that you have not enough protein synthesis, but that the people who believe in the protein synthesis being important think that a slower rate of protein synthesis produces less misfolded and protein aggregates, and that could be protective and make you live longer. People who believe in metabolism have another reason why changes in metabolism by mTOR would be important. And people who believe in autophagy think that mTOR going down produces an increase in autophagy and you have more recycling and therefore you live longer. You can make arguments for any of the downstream effectors of mTOR being the reason why reducing mTOR makes you live longer. And it may be, I mean, I think what's needed are effector mutants of mTOR, which have different inability to affect one pathway or the other.
All right. Well, I want to thank you, Lila, very much, and Richard and Toren for a most extraordinary, informative afternoon. Next week is drug addiction, habituation, drugs and food. So you're more than welcome. We look forward to seeing you.